Okay, I know we left off on the next slide, but I'm gonna reiterate this real quick. Do the next slide again, and then go from there. This slide starts the second line of defense mechanisms, and it starts talking about the main players, at least some of them, the macrophages. So macrophages and their phagocytic characteristics, the fact that they can phagocytose the bacteria, is a component of the second line of defense mechanisms. We talked about some of these white blood cells reside in the blood, specifically the monocytes, but once they enter the tissue, for example, monocytes are exclusively in the blood, but once they move into your tissue, they actually become a different cell called a macrophage. And these cells that live within tissue, once they reside within the tissue, they can live within the tissue, are together called histiocytes, the cells that associate with tissue. And the name of the macrophage is based on the tissue they're associated with, like the alveolar macrophages, those would be lung. Kupfer would be the liver. The dendritic, the macrophages that live in your skin are called dendritic cells. So those are just names for them, but they're all basically macrophages, and they are involved in this process, which is the second line of defense mechanism, phagocytosis. Number one, they will recognize that bacteria is not a host cell, because what does it lack? It's a social security number. What is that?
MHC1.
MHC1. Very good. So remember I said your MHC1 on the surface of all your cells identify you for you. When it comes to your white blood cells, how is the white blood cell going to know that that's you? It looks for a surface receptor on all of your cells except your circulating red blood cells, called MHC1, which stands for Major Histocompatibility Complex 1. And that's what makes you who you are, or recognizes who you are. And my MHC1 will be different than your MHC1, and so forth. So we have to take in consideration the MHC1s being expressed during a tissue transplantation, because if my MHC1 is not matching yours, my immune system is going to attack your tissue that you're trying to donate to me. So that's a side issue. So your white blood cells will recognize you for who you are by looking at your MHC1. So the bacteria does not have MHC1, luckily. So that's one reason, or that's one way your white blood cell is going to recognize that these are bacteria. They don't have MHC1. On top of that, they have their own calling card saying, hey, yeah, you're right, I'm bacteria. That's the pathogen-associated molecular patterns, PAMPs. On the surface of bacteria, you're going to have bacterial-associated proteins and maybe the cell wall, and see that, the cell wall and the outer membrane. These basically say, oh yeah, you are bacteria. I knew it. You didn't have MHC1, and you have these, you're bacteria. So it's a classical signature of, yes, I am bacteria. So then what happens is that your white blood cells has another receptor called the pattern recognition receptor that recognizes the PAMPs. And that's how your white blood cell attaches to the bacteria. It's through a PRR-PAMP interaction. So basically what your white blood cell is doing is grabbing onto the bacteria using the PRRs, those are on host cells, and the PAMPs, which are on bacterial cells. Now that I have grabbed onto it, I'll wait until I grab on a couple of them and then engulf all of them all together. The engulfing is the phagocytosis part. So what happens temporarily, at least, the bacteria is now in a vacuole called the phagosome, and what's coming is the lysosome with the hydrolytic enzymes in there, and they're gonna fuse together. So now once they fuse, this is phagosome, this is lysosome, and when they fuse together, this structure is called the phagolysosome. So now the bacteria is going to start feeling the pain. The pain of what? The pain of the hydrolytic enzymes. We told you before that lysosomes have hydrolytic enzymes that are very active in basically breaking macromolecules apart. And what's a bacteria? It's just a collection of macromolecules, just like you are. So no bacteria can survive the attack of the hydrolytic enzyme. Oh, that sounds like a good movie. The attack of the hydrolytic enzyme. Anyway, so once it all breaks down, it's now dead and it gets released. This part here is basically saying, okay, now I have this garbage, I'm gonna get rid of it. Does anyone know the name of this one right here? The release of things from the cell? Exocytosis. Very good. So that is one type of second defense mechanism. One of the PRRs on the surface of your cell is something called a toll-like receptor. This receptor here is on some of your, it's basically on a macrophage or some of your white blood cells, and it literally looks like this until it grabs onto a PAMP, a pattern-associated molecular pattern, a pathogen one. So once it grabs onto a piece of that PAMP, the structure of the PRR changes, and now it looks like this. See, that's why, remember I told you the full outer membrane of gram-negative bacteria does not hurt you. Because I managed to say that it's too big. No one asked me yet, but hopefully you'll realize it's too big for what? It's too big to bind here. So you really think that the PRR receptor, the toll-like receptor, is gonna attach to an entire outer membrane? It's too big, doesn't fit. But the toll-like receptor can easily attach, like we said before, to a piece of the outer membrane. Does anyone remember what this is called? The whole thing, of course, is the lipopolysaccharide outer membrane. But once it breaks apart into pieces, what do we call the piece? See? No?
It's very toxic.
It is very toxic in larger amounts, but it's toxic. It's toxic in larger amounts than the other toxic one. Not the exo, the endo. It's an endotoxin. Remember I said an endotoxin requires that the bacterial cell, which is gram-negative, dies, lyses. Because when it lyses, it breaks the outer membrane in millions of pieces. The toll-like receptor binds to one of those million pieces, not the entire outer membrane. It's too big. It's too big to fit in here. How am I gonna fit it in here? But a little piece of it can easily fit and cause the receptor to change form and activate the macrophage. Once the receptor arms, once it goes like this, it activates the macrophage. Activates the macrophage to do what? To secrete things like chemicals called cytokines, interleukins, inflammatory mediators. Basically, it sends the alarm signal. Alarm, 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 I found something. I found an endotoxin. So the bad guy must be around somewhere. Go find him. And that's the inflammation that we all basically feel or suffer from, at least temporarily, until the problem is resolved. Meanwhile, of course, it'll also be doing phagocytosis. So it phagocytoses while it's sending the alarm. So it's fighting a fire and at the same time calling for help. It's amazing how these things work. Okay, so that was a specific example of a PRR, the toll-like receptor, and how it works. Another second line of defense we're all familiar with is inflammation. If you ever step on something or if something ever bites you and all of a sudden it starts turning red, you feel the heat, pain, that's inflammation. These are the classical signs of inflammation: redness, swelling, pain, and heat. Like you see here, obviously something may have bit this person on the foot and your body is reacting to it. That inflammation is a good thing, and we'll show you why. Acute inflammation is good. It helps you, actually. Chronic inflammation is not good. To have inflammation for a long time, that is not good. You're gonna feel the pain of that. Not today, not next year, maybe 10, 15, 20 years from now, when other things start happening to you. So you don't want inflammation to hang around for a long period of time. So that's gonna cause other diseases. But acute inflammation at the time of an insect bite or stepping on a nail or whatever, that is something that is good for you because it's fighting the pathogen. So this is an example of what would happen. And you walk outside barefoot, you step on a nail or something. Obviously you broke the first line of defense, which is your skin. When the point of view of the bacteria gained the portal of entry. You see how we put the information together? So now the bacteria has gained entry and it's gonna start multiplying, adhering to your tissue, multiplying, avoid host defense mechanisms as much as they can. But you have your own tricks up your sleeve here. You're not gonna just sit there and take it. Your resident cells will start attacking the bacteria. The ones nearest to the injury will start working for you, I hope. But at the same time that they're working for you, they're sending the alarm signal. Alarm, alarm, we have a problem. Come and help us, please. And that's what sets off the inflammation, the alarm signal that's being sent, and the new players coming into the site of injury is the one that's going to cause the inflammatory response, which is a good thing in this case. Because at the end of inflammation, the goal is to do two things. Number one, get rid of the pathogen, which is good. Number two, fix the damage caused by the pathogen and by the needle or the nail or whatever. So, the alarm signal sent by the white blood cells is something called the chemotactic factors. The alarm of the white blood cells are the chemotactic factors, are called chemokines as well. So I'm standing here fighting bacteria and I'm sending an alarm signal in the form of a chemical to attract you to come to the site of injury. A chemical that attracts cells to move to the site of injury. What did we call that? The movement towards a chemical. Chemotaxis. Very good. So that's one component. Another component is that they will also release what is called vasoactive factors. Vaso means blood vessel. So factors that affect the blood vessels. To do what? We'll show you. But these factors affect smooth muscle cells of the blood vessels as well as the endothelial cells. So they affect them in a certain way to achieve a certain goal. And we'll show you what happens. So these cells, once they sense that there's a problem in this area, they will release both chemotactic factors as well as vasoactive factors. The chemotactic factors will be involved in moving cells towards the alarm signal. It's sort of like a fire truck driving around, hearing a fire alarm. The truck will drive towards the fire alarm. That's what's attracting the fire truck. The sound of the alarm is similar to that. But meanwhile, these cells will also release factors that affect smooth muscle cells on the blood vessels as well as endothelial cells. So, number one, the chemotactic factors attract white blood cells to the site of injury. See, What would normally happen is if I was a white blood cell in the blood vessel, if I don't hear an alarm, I just keep moving in the vessel. The blood pressure is pushing me through the blood vessel. Well, it's okay because there's nothing going on. I don't hear the alarm, so I just pass right by. Versus there's damage right here and the signal is coming from here. Alarm, alarm, come and help us. If I'm a white blood cell, it would be really, really bad to pass right by that. I would not be doing my job. The signal is going to attract me towards it. Well, the alarm is coming from the tissue, but I'm in the blood vessel. So I will show you in another slide, I will have to leave the bloodstream and enter the tissue to do the fight. And we'll show you what happens there. So that's one thing. I'm not going to pass by a chemotactic factor alarm because that would mean I'm not doing my job. So the chemotactic factors released by these white blood cells will attract another white blood cell towards the site of injury to help. But then it realizes I'm not getting enough helpers. I'm not getting enough fire trucks to come to the scene. Maybe because the road is too narrow. What if I widen the road? I'll get more fire trucks there. Do you see what I mean? That's the vasoactive factors. What if I cause your blood vessel to dilate? Oh, I'll bring more cells then. So it's about cells reaching the site of injury, and it's also about a large number of cells reaching the site of injury. So the alarm tells the cells where the problem is, but the vasoactive factors dilates the blood vessel to bring all the help it needs all at once because there's no time to waste. It's amazing how that works, and it's like really quick. If you ever been bitten by something and then all of a sudden within like 20 seconds after that it's already red and hurting you and all that because it's really, really quick. The interesting thing is we learned not too long ago in this chapter that blood is not just a cellular component. Blood is also a liquid. Remember that? So wherever the cells end up, so will the liquid component of blood. That's why it swells. Okay, I'm sending a signal, widening the road or the blood vessel, dilating the blood vessel to bring more cells. But with the cells comes the fluid, the plasma. So it's going to swell. But even that has a function. Even the swelling, the delivery of the fluids into the site of injury has a function. It's amazing. The function of the fluid in the tissue is basically to dilute out the toxins. See, these toxins released by the infection, by the bacteria, it's damaging the tissue. So while the bacteria is there, the toxins are damaging your tissue. So I know I'm sending cells to fight the bacteria, but I should also try my best to contain the damage. While I'm fighting off the bacteria, contain their effect, minimize the damage, because I'm gonna have to fix it later. I'm not gonna kill the bacteria while they're damaging my tissue, kill the bacteria while no, I'm gonna start killing the bacteria and at the same time dilute out the toxins to minimize the damage to your tissue. It's amazing. Even the swelling, the slight edema, the swelling, has a function. It basically dilutes out the toxins so they're no longer effective, giving you a chance to kill the bacteria, at the same time minimize further damage. It's amazing how that works. Once all the bacteria are gone, it also fixes the inflammatory response, also starts fixing your damaged tissue. So that's inflammation, and these are classical signs of inflammation. Every time you go through that, that's what will happen. Now, of course, you feel the pain from the swelling, it pushes on nerves, and that's the pain associated with it. The redness is the high level of blood reaching the site, as well as the heat. Wherever blood goes, that's the area that's going to be higher in temperature, of course. So it goes all hand in hand. Now, what about these white blood cells that are originally in the blood vessel and get the signal, the alarm, 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 come over here and help us? Well, they're going to have to leave the bloodstream and enter the tissue. So what happens is that initially they don't have that in the slide, so you might want to add it. Initially, they have to start rolling. So once they hear or they get the signal, the alarm, they literally start rolling to slow themselves down. And then they start marginating. That means attaching to the surface, the inner surface of your blood vessel. Luckily, it's only one cell layer thick. It's the endothelial there. It is one cell layer thick, and it's literally able to squeeze between them and enter the tissue. The squeezing between them is called diapedesis. So, for the white blood cells to eventually enter the tissue, it has to go actually through three things. One of them is not on the slide. The first thing is rolling. The next thing is margination. And the fourth thing is, the third thing, not fourth. The third thing is diapedesis. So rolling, margination, and then diapedesis. Then it's in the tissue and it starts fighting, helping you. Questions on any of this so far before we go to the next one? Obviously what we're doing is we're showing you all the different second line of defense mechanisms that are always working for you. We talked about phagocytosis so far, the PAMPs and the PRRs. We gave you an example of the toll-like receptor as an example of a PRR. We talked about inflammation, gave you the signs of inflammation and how the chemotactic factors work, how the vasoactive factors work, and how cells leave the blood vessel and enter the site of injury by rolling, margination, diapedesis. The next second line of defense mechanism, if you don't have any questions then, is fever. We're all familiar with this. We all suffer from pathogens and so forth, and we develop a fever. Fever, in this case, is a good thing. Low-grade fever, 37.7, 38.3, moderate and high. I think I usually, basically, at least for myself, and I try to do it with my children, but my wife always treats at 37.8. But I like to wait until 40, then I start treating. Because these actually are actually good. Fever is actually good. But some people, once they see the temperature go up, they panic and they start treating the child, which is actually not good. Not as soon as the temperature goes up, you treat the child, because there's actually a benefit to fever, and we'll show you that. But once you start hitting 40, 41, there's still benefit, but then your child is suffering meanwhile, so you need to treat that for sure. So this area here, I'm okay with it. Maybe here I start thinking, okay, but by the time we get to here, that's for sure, you need to treat that. Now, there are things that stimulate fever. Exogenous factors that stimulate fever are your obvious bacteria, viruses. Endogenous factors that stimulate bacteria are some of these chemicals or factors released from macrophages, from your white blood cells, as part of the inflammatory response as part of them being stimulated when they capture something foreign. They send chemokines and cytokines and things like that, and that causes your temperature to also go up. But that's endogenous. That's your cells releasing these factors in the process of them fighting off the pathogen. So you can develop fever from either exogenous agents or endogenous, or pyrogens are fever generating or heat generating agents, pyrogens. There is benefit to fever. The first benefit is, of course, with heat, we denature proteins. So bacterial proteins can be easily denatured, and then of course they don't work anymore for the bacteria and they die. We are a little bit more complicated than that. So a lot of our proteins can withstand a slight increase in temperature, unlike a lot of the bacterial proteins can do. So we can tolerate a somewhat of an increase in temperature with no problem. But don't push it, because if the temperature keeps going up, even our cells will be affected by it. So that's the advantage that you see. Bacteria are more susceptible to the increase in temperature than our cells are. So we can kill them before we start hurting ourselves. But if you keep pushing it, then both the bacterial cell and our cells are now hurt by the temperature. That's when you have to treat the patient. The other advantage of low-grade fever is this. Look at that. It stimulates your immune system. So if you treat early, you're going against the benefit of fever. Low-grade fever helps protect you by simply stimulating your immune system. So you treat too early, you're losing this effect. I tried to tell that to my wife, not successful, right? So she still treats my child. As soon as she feels a little bit of a fever, she goes and treats him right away. It's understandable. All right. The next example of a second line of defense is you may have heard it before, and I think you did. I hope you remember interferons. Interferons are released by a virally infected cell. So one of your cells gets virally infected. That cell will secrete an interferon. It's like saying to the neighboring cells who are healthy, hey, a virus attacked me. Go protect yourself. I'm doomed. I myself am doomed. As a last selfless act, before I die, I'm going to send an alarm telling you that there's a bad guy in the neighborhood. I mean, this is amazing. If I'm a cell and I get virally infected, I know I'm doomed. Now, this is a challenging question. Who's coming to kill me? I'm a host cell. I'm virally infected. One of your cells, one of the cells from the host, is coming to kill me. Which cell is that?
A white blood cell?
Which one?
Oh.
A white blood cell.
A neutrophil.
No.
Are you viral?
I am a host cell that's been virally infected. So I'm one of your cells. But I have a virus in me. I'm no good for you anymore. So another white blood cell of yours is gonna go there and say, commit suicide.
Is that the eosinophil?
Not the eosinophil, that's for worms and fungus.
Okay, basically.
You're on the right track there.
Yes.
Say, not basophil.
Neutrophil.
Say,
Neutrophil.
not neutrophil,
B cells.
not B cells. B cells release antibodies.
Monocytes?
Yeah, oh, you guys are naming all of them except the one I want.
Lymphocytes?
Which lymphocyte?
Oh.
Neutrophils.
See, B cell is a lymphocyte.
T cells.
T cells.
Oh.
You think I'm done with you? Which other cell too? There are actually two cells.
The natural.
Natural killer cells. Here we go. So watch this. A virally infected cell. Oh, this is a host. This is one of your cells. It's no good anymore. It has a virus. You don't want it anymore. Because it's now just a viral factory. It's no good for you anymore. So your T cells and your natural killer cells are gonna recognize that. Hey, you're infected. Kill yourself. We don't need you here anymore. Yeah.
Does it know that it's infected because of the interferons?
No. You'll see how. You'll see how. That's the next chapter.
Okay.
But before the T cell and the natural killer cell reach this cell, is gonna do this. Send the signal out. Hey, you guys. Jeff, Megan, Jocelyn, everybody, protect yourself. I'm doomed. But you need to protect yourself. The bad guy is in the neighborhood. That's how we minimize the effect of the virus. This interferon is like an alarm signal to the healthy cells to prepare themselves. He's gonna come ringing the doorbell. Remember all of that? He's gonna absorb, penetrate. You remember all of that? The animal viral infection cycle there. No one warned this cell, so the virus absorbed, penetrated, uncoated, and infected the cell. But this guy, knowing that he's doomed, he sends this interferon signal to tell the neighboring cell, change your gene expression pattern. Produce antiviral proteins. So when he comes knocking, surprise, you're not touching me. I prepared for you. Do you see that? Because I got the message. It's amazing. Later in the next chapter, meanwhile, this guy here is going to look for it in the next chapter. Is going to take a piece of the virus and take it to the surface of the cell and do this. This cell is going to take a piece of the virus, bring it to the surface of the cell, and do this with it. Here's the virus right there. Yeah, I'm virally infected. Here's the virus. Here's a piece of it. And that's how the T cell, as the T cell is going by, it realizes, oh, you're telling me you're viral infected. Kill yourself. It's amazing how this works. And it will show you how it kills it. If it did not take a piece of the virus and stick it on the surface, how is the T cell going to know that there's a virus inside? So it will take a piece of the virus and send it to the surface, marking it for destruction. By who? By the T cells, and what was the name of the other cells? Natural killer cells. It's amazing how that works.
Does the signal only go to the nearby cells?
It will go as far as it goes, but it's in the neighboring cell. They normally don't, they get dumped into the blood. It's like in the tissue, in the neighboring. Yeah. So it's like a local signal.
So then does the virus stop traveling after you can't attack the next cell?
Well, what happens is, remember, viruses are tissue specific. So if this is a liver, the problem is going to be in the liver. So as long as the neighboring liver cells are getting the signal, they're going to protect themselves, and eventually the ones that are infected will be killed, and then you wash out the virus, and hopefully you'll be fine. Yeah. Okay, another second line of defense mechanism is called the complement system. I need your attention, please, with this one. The complement system is a collection of 30 proteins that are circulating in your blood right now. 30 different kinds. And they're named C1, C2, C3, C4, and some of them are C3A, C3B, and all. So C for complement with a number and or a number and a letter. So about 30 of them, and they're in your blood right now. Hopefully for all your life, because they're very important. What you need to do when you get an infection, the complement system is gonna recognize that's bacteria. And I need to do something about it. So how do they recognize bacteria and how do they activate the complement reaction? Well, they first have to recognize its bacteria and start assembling on the surface of the bacteria. Remember, there are 30 different proteins. They're not going together. They're in separate, they're soluble proteins. They go on their own. They only start coming together when they're fighting the pathogen. So the initiation of the process was discovered to be like this initially, and then we discovered two other ways. So there are three ways of initiating the process, starting the process. And you'll understand what the process means when we finish this off. So these are called initiating complexes. You have, for example, C1 recognizes the bacteria that's been marked by an antibody. So we'll talk about antibodies in the next chapter, but antibodies attach to the surface of pathogens, at least in some cases. That marks them for bad guys. See, if I have a bad guy like this marker here, and if I'm C1, I'm one of the complement proteins, this guy is traveling right next to me and I'm like, I don't know who that is. And it goes right by me. And I do nothing. I mean, my job is to grab onto this guy and kill it. But no one told me that this is a bad guy. It's not marked for destruction. How do we mark this guy? We attach antibodies to it. So the B cells will produce antibodies that recognize, oh, that's bacteria. The antibodies, which are Y-shaped structures, they will bind to the bacteria, marking it for destruction. And that's what you're seeing here. That's an antibody attached to the surface of the bacteria. Now, when that guy goes by a C1, the C1 will attach to it because it's been marked for destruction. So, without the antibody, this bacteria will go right by the C1, nothing happens. But you mark the bacteria with the antibody to tell the C1 protein, yeah, that's a bad guy, kill it. How do you kill it? You start by producing this, what's called initiating complex. You're producing a bridge between the bacteria and the C1. The antibody is the bridge. So you got the bacteria, the antibody, and then the C1. Without the antibody, the C1 has nothing to grab onto. So the antibody serves as a bridge. Now, in effect, the C1 is now grabbed onto the bacteria, and a cascade of events begin after that. And it's a phenomenal cascade of events, and thank God I'm only touching the surface of it. To learn all the events involved in that would require like 3 or 4 or 5 lectures. And it's phenomenal. So what we're doing here is initiating the cascade by simply producing the bridge or the connection between the C1 and the pathogen. And the bridge is the antibody. Now it grabbed onto it. Now a whole bunch of events are going to happen that will ultimately kill the bacteria. Well, you can also do the same thing, but using what's called the lectin pathway. This is a different initiating complex and it recognizes certain sugars called mannoses that are on the surface of microbes. So the classical pathway recognizes the antibody that's been bound to the pathogen. The lectin pathway recognizes the mannose on the surface of pathogens. And we thought that was enough, right? There's a third one. The alternative pathway is an initiating complex that recognizes the N-acetylglucosamine, which is a component of the bacterial cell wall, the peptidoglycan. It is one glycan of the peptidoglycan part. The N-acetylglucosamine is one of the glycans. The other one is the N-acetylmuramic acid. So it recognizes the cell wall. For sure it's bacteria, because only bacteria has peptidoglycan cell walls. And it really doesn't matter whether it does this, or this. The ultimate consequence to the bacteria is the same, and that is, of course, death to the bacteria. And I'm going to try to be as brief as possible, knowing that this is quite involved. And if you want to watch a video on it, you can go to this one here. But regardless of which initiating complex was produced, they eventually all feed into activating the C3 complement. Remember, the complement is about 30 different proteins. They're all labeled, named C with a number. So they will all eventually cause the activation of the C3 complement. Now, you gotta understand what I mean by the word activation. In this case, it means taking C3 and cleaving it into two pieces. A small piece and a larger piece. So if you notice, C3 will become C3A and C3B. It's sort of like this. Here's C3. To activate C3 is equivalent to doing this. Like that. C3A, C3B. I use my markers a lot. C3A, C3B. That's what I mean when I say activate. And that's what I will mean when I say activate with the other ones too. Cleavage. It means you're cleaving it. C3A is going to do one thing, and C3B is going to go and do something else. Everything has a job. Meanwhile, you're also activating other complements, proteins. So these guys here will be involved in the inflammatory response. It will initiate inflammation, which is a good thing. Remember that. It helps you fight the invader. These guys here will be involved in something called, you'll hear it next chapter too, but you'll hear it here, opsonization. That is such a fancy word for something simple. Enhanced phagocytosis. So the cells that phagocytose, they do it at a certain rate, with certain success rate. But when you opsonize them, you make them super phagocytes. Do you see that? There's a difference between a phagocyte and a super phagocyte. You can tell, right? So how do you change a phagocyte into someone that it can really do the job? Phagocyte, it's called opsonization. Opsonization is enhanced phagocytosis. That's the best way I can explain it there. So every piece of the complement system is doing something. Inflammation, to do this. To do, which one? Phagocyte, yeah, this one. Enhance inflammation so we can be a better machine there. Bring more players to the site of injury and start really fighting. Increase or enhance phagocytosis so we can really kill them. But those, believe it or not, are side effects of the complement system. The real job of the complement system is this one. every time you hear about the complement system, if you learn about it in detail, which you are, you're learning about it, you will remember this part. The membrane attack complex. The complement system is known for basically putting holes in the membrane of bacteria. They put a hole in the bacterial membrane. So what happens? Everything inside the bacteria leaks out. What do we call that? When the contents of your cells leak out and the cell dies.
Lysis?
Yeah, lysis. We call it lysis. And I'm not talking about one hole. I'm talking about thousands or millions of holes on the membrane. No bacteria can survive that. So this is what the complement system is really known for. The formation of the membrane attack complex, MAC. It's a bunch of these complement proteins, specifically C9, coming together, sitting in the membrane, like this. If this is the cell membrane of bacteria, the C9 complement will come and sit here, 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 creating a pore. And it will do the same thing here, for example, creating another pore, and another one, and another one. And the contents of the cell leaks out, and we call that lysis, and it dies. That's what it's really known for. The formation of the membrane attack complex. But as a side shoot, it will also enhance phagocytosis, which is called what? Opsonization, and stimulate inflammation to bring more players to the site of injury. Because when your immune system goes to fight or goes to war, it doesn't just bring guns. It brings guns, tanks, airplanes, knives, bats. It brings everything. The kitchen sink, you see? It fights with everything. Because what's the consequence of it losing the fight? You dying. So you're like, thank you for doing all of that for me. Thank you for just not fighting like this. Thank you for doing everything in your control to help me get rid of this pathogen. It's amazing how that works. Another second line of, we've got two more, we're done here. Another second line of defense, this is an easy one. Iron. Iron is very important in certain aspects in our body. There, for example, hemoglobin needs iron to form the three-dimensional structure and to be able to grab onto the oxygen and all this. So without iron, you're going to have a disease called anemia. At least one type of anemia is caused by iron deficiency. So that's where we hide the iron, technically, inside red blood cells. So when you eat food with high in iron, it's for the purpose of using it. And one very good place of using the iron is in hemoglobin. So technically the iron is hidden inside your red blood cells. It's being stored in certain areas in your body and in your fluids and so forth. So we know how to hide the iron away from bacteria. Because it's like saying, it's like those kids that say, it's mine, this is mine, don't touch it, it's mine. So that's a good thing in this case. If you talk to bacteria and say, don't touch the iron, this is mine, that's good. Because if the bacteria needs the iron to survive, by you taking the iron and saying, it's mine, you're killing the bacteria. Are you catching that? It's an indirect effect. If the bacteria needs this to survive, and I do this, don't touch it, it's mine, then I kill the bacteria. That's similar to this. It's amazing. But then you have these bacteria that do this. They wait. They're so patient. They wait, and they wait until you do this. Let it go. They're like, let it go. I dare you to let it go. Just let it go. Let it go. Let it go. I got cytophores. So some bacteria produce cytophores. waiting for you to let go of the iron. Once you let go of it, they grab it and they say, now it's mine, and you're not getting it back. So what do you think bacteria that produce cytophores, will they survive? Of course they will. They figured out ways to get to your iron. All they do is say, just let it go. And I got something that will really grab onto your iron. And even if you try to take it away from me, look at that. I hang on to it more tightly than you can. So you might be holding the iron like this. The cytophores produced by bacteria are holding the iron like this, trying to get it now. You can't. So bacteria that produce cytophores to grab onto your iron more tightly than you can grab onto your own iron will survive. So now you'll have an infection. So it's kind of like an indirect effect. If you can hide the iron from bacteria, that's a second line of defense mechanism, because the bacteria that need the iron won't survive unless they're producing cytophores. And they're waiting for you to just let it go, and I'll get it. So when will we let go of the iron in this example here? Well, the red blood cell has a life cycle. If I remember right, it's about 120 days. At the end of the 120 days, the red blood cell will burst open, release the hemoglobin and the iron as a means of recycling things. Well, that's when the bacteria will grab onto that iron and say, yeah, thank you for that, but you're not getting it back. It's amazing, again, how this works. It's a war between you and the pathogen. And may the best person win here. The last thing, very easy here. Do you remember that I said to you guys that some white blood cells have granules? Do you remember that? The granulated cells are the what? Never eat bananas. The neutrophils, eosinophils, and the basophils. Those are the granulocytes. Well, some of the granules have chemicals in them, some of them are like these, that, of course, kill bacteria. So the list of chemicals is unbelievable there. Different granules have different chemicals in there, and different cells have different kinds of granules. But the overall effect of that is what they do. They secrete these chemicals in their attempt to kill the pathogen. Some of these are antimicrobial proteins. They're very small, technically more like peptides, 12 to 50 amino acids or so. They fit in the cell membrane of bacteria, and they disrupt the cell membrane for bacteria. And as soon as you disrupt the bacterial cell membrane, the consequence, of course, is lysis. Anything that disrupts the cell membrane, whether it's a surfactant or the complement, the MEC, the membrane attack complex from the complement system, or these guys, the antimicrobial peptides, the ultimate goal is, of course, lysis of the bacteria. So we discussed a whole array of second line of defense mechanisms. If we review them in our mind there real quick, if I can remember all of them, phagocytosis, remember that? That's when we started talking about the PAMPs and the PRRs, and we gave you an example of one of the PRRs, the toll-like receptor. What did we follow that by again? Inflammation, yeah. Inflammation was the next one. You need to know all the classical signs of inflammation and the chemokine factors and the vasoactive factors. The benefit of the edema, the diluting of the toxins. And the fact that at the end of inflammation, you're going to have the pathogen destroyed, but you will also fix the damage at the end of it. The movement of white blood cells from the blood vessel into the tissue involves rolling, migration, and what's the third one? The squeezing in between? Diapedesis. Very good. Now, what was the next one I talked about after that? Was it fever? Can't remember the order of them. Is it fever? Yeah. Fever is the next one. Remember, low grade is not too bad. It's actually beneficial in denaturing bacterial proteins, in effect killing them. It's also beneficial in the fact that it stimulates your immune system. But once your temperature goes too high, you must treat the patient, because it can also be fatal to the patient. So that's fever. And then we talked about interferons, am I right? Interferons are these alarm signals produced by virally infected cells. They know they're already doomed. Their life is about to end. But as a last resort, let me warn my neighbors, my healthy neighbors, to protect themselves from the virus. Because the virus is coming. It's going to absorb, penetrate, and try to infect my neighbor's cell. But surprise, got the message and prepared. It's all through gene expression. So the healthy host cell, once it gets the message, it will change gene expression to produce antiviral proteins. And now, surprise, I knew you were coming, and you destroy the virus. Remember that you'll see that in the next chapter. Meanwhile, the virally infected cell will be killed by the T cell or the natural killer cells. And you'll see how that happens in the next chapter. Then what did we say after that one? The fever, the interferon, and then the complement system. Am I right? Remember, there are 3 initiating complexes. I don't care which one initiates, the ultimate effect is the same, and that is basically lysis of the cell through the formation of the membrane attack complex. But you also get increased inflammation, which is a good thing in this case, and you also get enhanced phagocytosis, which is called opsonization. I don't know where to get that one. Opsonizing. Then what did we say after that? After the complement system? The iron, yeah, the siderophores. The siderophores is an indirect effect there. The iron, the fact that you're hanging on to the iron and the bacteria wants it, that's a kind of like an indirect effect. You're killing the bacteria by not giving it access to the iron. But once you let go of the iron, it will grab onto it much tighter using its siderophores. And then the last one, the antimicrobial peptides, they interrupt the arrangement of the phospholipids in the bacterial cell membrane, and it causes the bacteria to lyse.